In this video, we're going to talk about digestion of carbohydrates. Like we said in the introductory video, we're going to divide our discussion of digestion into three parts. We'll do carbohydrates in this video, we'll do proteins in a separate video, and we'll do our lipids in one more video. So for this, we're going to say you were eating, I don't know, some bread, you were having a sandwich, maybe some, I don't know, pizza crust, whatever. And it's going to start with the bread, the pizza, the carbohydrates, basically, going into your mouth. So when they go into your mouth, we start two things. We start chewing them. So that's mechanical digestion. We also start breaking them down with some enzymes. But before we can talk about breaking them down with some enzymes, we have to talk about what they are and exactly what the structure is. So primarily what we're going to get from bread, from pizza crust, from pasta, whatever, are amylopectin. That's our branched carbohydrate. We're going to get some amylose. So the first thing we have to do is we need to start talking about the bonding in those molecules. So we'll start having that conversation about bonding in a molecule of amylose. So amylose is a whole bunch of single sugar units attached to each other. So what we're going to talk about is how they're attached. So if I start at the oxygen and start counting clockwise, I have carbon 1, 2, 3, 4, 5, and carbon 6 sticking up out of the ring. Again, carbon 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And so on as our chain continued. So when we look at this, what we can see is we have a carbon 1 of one sugar attached to the carbon 4 of another. Carbon 1 to carbon 4, carbon 1 to carbon 4, and so on. So what we call that bond is an alpha 1, 4 glycosidic bond or linkage. So remember, GLY, GLU, anything, you're starting to think about carbohydrates. So carbon 1 to carbon 4 through an alpha 1, 4 glycosidic bond. So when we look at our branched molecule, our amylopectin, we have our same numbering. I forgot my carbon 6. And when we look at the linear part of the chain, we have exactly the same thing in terms of bonding. A carbon 1 to a carbon 4, 1 to 4, 1 to 4, 1 to 4. So those bonds are our alpha 1, 4 glycosidic bonds. But to make a branch, we need to be able to connect a third molecule to two that are already connected. So we look at this sugar, we see carbon 1 is occupied, it's attached to something. Carbon 4 is occupied, it's attached to something. Now we're going to use carbon 6 to attach to another carbon 1. So here we have carbon 6. I didn't do a very good job of showing it sticking up. Carbon 6 attached to another monosaccharide's carbon 1, or another sugar's carbon 1. So what we have going on there is we have a 1,6, a carbon 1 attached to our carbon 6. So again, not a great job of drawing it, but what that is an alpha 1,6 glycosidic bond. And that's what gets us our branching. So linear molecule or linear part of the molecule is attached through 1,4 bonds, alpha 1,4. The branches are connected with 1,6 bonds. Then we go back to 1,4, 1,4 and continue that branch until we get to another branch point. So that's the structure of our amylose and our amylopectin. We have some other molecules we'll get from the bread or the pizza crust or whatever. We'll get some cellulose, which you can't really break down. It's the fiber that helps with digestion, stuff like that. So we're going to focus on the amylopectin and on the amylose. So what happens is we have the salivary glands. We have extrinsic glands. We have intrinsic glands. And they're going to release, well, the extrinsic glands are going to release saliva. In that saliva, we have an enzyme called alpha amylase. Alpha amylase is going to act everywhere from the mouth to first part of the stomach. So it'll start breaking down these big molecules, and it'll start in the mouth. We also have mechanical digestion going on in the mouth. We're going to chew in the mouth. And it'll continue through the esophagus and everything, all the way down to the first part of the stomach. The pH drops in the stomach. Alpha amylase can handle a little bit lower pH, but not that lower pH. So it'll work for a little while in the stomach and then only get so far. Alpha amylase probably breaks down 10 to 15% of our carbohydrates, or probably a better way to say that would be about 10 to 15% of our carbohydrate digestion is accomplished by alpha amylase. So we've got these two molecules, and I'm going to put another slide in just to have room to work, and we'll keep doing that. I have that amylopectin. This one, remember, is the branched molecule. I have amylose. This is my linear molecule. I have salivary amylase working on these. And it's going to break these down into three things. So remember, I have the 1,4 bonds here and the 1,6. I have the alpha 1,4s here. So what I'm going to get is three things. I'm going to get maltose. So my maltose is a disaccharide, 2 sugar units. So this one's my disaccharide. I'm going to get maltotriose. I'm also going to get an alpha limidextrin. I'm going to keep the branched part of the molecule. So these two, being more than 2 units, are oligosaccharides. So what you'll notice in all of our digestion stories is we're going to start out with large molecules, and we're going to sequentially break them into smaller and smaller pieces. So I broke the amylopectin and the amylose down into disaccharides and oligosaccharides. So into these smaller units. So I'm going to take these smaller units now and keep going. We did that up till a little bit of the first part of the stomach, stopped in the stomach. Now we're going to move into the small intestine. So in the small intestine, we're going to come in with our three molecules. We're going to come in with our maltose, maltotriose, and our alpha limit dextrins. And what's going to happen now? We're in the small intestine and really in all the parts of the small intestine, but we have the pancreas there. And the pancreas can release another amylase. And remember the adjective that sounds like a place in front of a molecule name just means where it came from. Salivary amylase, pancreatic amylase, things like that. So we're going to have a hormone called cholecystokinin that will stimulate the release of this pancreatic amylase. It'll release it into the small intestine so it can keep breaking these molecules down. Let's write down the cholecystokinin. See, I have it. Stimulates the release of pancreatic amylase, continues breaking down the larger molecules, really just gets us more of these. So as we go through the small intestine, we're getting more of these three molecules. That will continue as we go through. Then next thing we need is enterocytes. So the enterocytes, I had one pre-drawn here. The enterocytes, if you remember from histology, are the cells bordering making up the lining and the sides of the small intestine. So these are the cells that are going to be the big players in absorbing these molecules. They have kind of a wrinkled cell membrane. And remember we talked about wrinkled cell membranes mean we want the wrinkles to get more surface area. We want the surface area to have more enzymes, other proteins, whatever we need embedded in there. We need space for it. So we have that. The bigger surface area is a result of the microvilli. That's the name for the little kind of finger-like protrusion sticking out. We use that surface area for absorption, digestion, things like that. One of the things we have in that increased surface area in those microvilli is we have the brush border enzymes. So that's those things I drew pretty horribly sticking out. We're going to go through them one by one and talk about what they are. So we've got 4 types here. Actually, we'll go through and we'll talk about, well, we'll write them. We'll write them and go through and talk about them. So one's a lactase. We'll talk about that for breaking down lactose. We have a maltase. And you guessed it, breaks down our maltose. It's also known as glucoamylase. We have sucrase. We'll talk about it. And we have isomaltase. These two guys hang out together. So we have this enterocyte hanging out on the side of the small intestine, on the wall of the small intestine. And we're going to talk about what happens. So we have some molecules that we talked about. We have the 3 that we originally talked about. We have our maltose. We have our maltotriose. We have our alpha-limit dextrin. So we talked about those three and the structures of those three and everything. We also have lactose and sucrose. So we'll go through these one by one and talk about breaking them down. So what we're going to do is we're going to break down all of these into the individual monosaccharide units. Talk about that, how we do it, where we do it, things like that. So we're going to start with the lactose. So we're going to break lactose down into glucose and galactose. The enzyme that does that is lactase. So this is supposed to be my lactase. So what will happen is the lactose will go in there. I'll break down my lactose into a glucose. We're going to use G for glucose and a galactose. We'll use GAL for galactose. Lactase gets pretty important. You get lactose in your dairy products, and if you don't have lactase, you're going to have a lot of GI issues because you can't break down your lactose. Those are your lactose intolerant patients. So it's a pretty common thing. So it's something you will hear about and think about with your patients. Okay, the next brush border enzyme is maltase. So this one's our maltase. What this one's going to do is take maltose and maltotriose, which is really kind of sort of a whole bunch of glucoses hooked together, take those and break them down into individual glucose molecules. So we had the chain of 2, chain of 3. Now we're going to have the individual glucose molecules. Our next enzyme that I attempted to draw here is my sucrase and my isomaltase. So this one's supposed to be my sucrase. This one's supposed to be my isomaltase. So they're together, and they do a couple different things. So when I want to break sucrose down, that's where my sucrase comes in. It'll break sucrose down into glucose and fructose. So here, I'm going to get a glucose and I'm going to get a fructose. We'll just do an F for fructose. So it can also, sucrase can also break down these three guys as well. Then last one we're going to talk about is our isomaltase. This one's a little bit different. So remember with our alpha-limit dextrin, we still had the branch point. So the branch point was formed by that alpha-1,6 bond. So this enzyme, our isomaltase, breaks the alpha-1,6 bond. That's the difference there, and gives us still glucoses, but breaks that bond to get the glucoses. So now we've got everything broken down. We have our glucoses, our galactose, more glucoses, and a fructose. So I've got a few enterocytes drawn here. So we'll go to a new enterocyte to have room to work. So I have glucose, lots of it. I have galactose, and I have fructose. So we use G for glucose. We have all our little G's. We had our galactose. We did a GAL, the G, and we had an F for our fructose. So we've broken everything down into these individual units? Well, fructose is a little special. Fructose has its own personal way into the enterocyte, and it's a transporter. It's called glucose transporter type 5. Short form is GLUT5. And that's how fructose gets in. So that gets fructose into our enterocyte. Then we have another story for getting our glucose and galactose in. So this story involves sodium. So we have some proteins, we have some cells that can transport some ions, and we have a story with that. We're going to have in these cells, we're going to have sodium leaving the cell. And for every 3 sodium that leave the cell, 2 potassiums go in. What happens when that happens, though, is we end up setting up a gradient, because we've got the rest of the small intestine and everything here. And what we've done by all that sodium leaving is we end up with a higher sodium concentration outside of the enterocyte. So what happens because of that higher sodium concentration and this gradient is, that color's not good, we haven't used green for a while, is the sodium wants to move back in. So the sodium is going to go back into the enterocyte, and with it, it's going to take some other stuff. It's going to take glucose with it. It's going to take galactose with it. So the sodium will go in with the glucose, with the galactose. So there's a name for this transporter. It's the sodium glucose transporter. So we use that, and we got our sodium back in. So we're balancing out our sodium again. Not really the point of what we're doing. We got our glucose in, we got our galactose in, we got our fructose in. So they're in the enterocyte. The enterocyte is their ticket into the bloodstream. Oh, and I forgot up here, we use ATP while doing this also. So we're trying to get all of these things into the bloodstream. So we have another transporter. We have what's called, running out of colors. We'll go red. We have a GLUT2 transporter. That's supposed to be these things. And it's not specific to anything. So what the GLUT2 transporter is going to do is going to take the glucose, take the galactose, take the fructose, and all 3 of those can go use the GLUT2, go into the bloodstream. So that's how we get all of these things into the bloodstream. So next trimester, when we're talking about metabolism, we're going to be talking about, you have glucose in the bloodstream, it goes into the cell, and here's how it produces energy. You have fructose, you have galactose in the bloodstream, get them in the cell, and here's what happens. So this story will end here with the bloodstream. We're going to do the same thing with the lipids, same thing with the proteins, how we get them into the bloodstream. Then next trimester, we'll pick up from there.